So I have been at Colorado State, as Bob said, about 3 years now, just a little bit over. It has been an adventure. As I was saying, a lot of people think that BioMarc is a startup. We're not. We've been around since about 2008, 2009 era. We are a contract development and manufacturing organization. So we are almost exclusively funded by outside contracts.
Excuse me for just a minute, John. We're still just seeing that. There we go. Now we're seeing
I was
that.
killing time.
Oh, okay. Just wanted to make sure you didn't think you had your slide deck up.
Yep. So can everybody see these now and your screen is full?
Yep.
Wonderful. This is the second virtual conference I've done this week, so I'm becoming at least familiar with all kinds of different presentation platforms. Okay, so I'm the director of BioMarc. I'm also an affiliate professor of immunology in the Department of Microimmunology and Pathology at the College of Veterinary Medicine and Biomedical Sciences at CSU. And so while we're not a startup, we do deal with startups in our contracts, as I was beginning to say. And let's see, got to advance it over here. Okay. So a little bit about BioMarc, and as Bob said, we're the Biopharmaceutical Manufacturing and Academic Resource Center. So that gets us into a little bit of the university mission, more along the land-grant university of research, teaching, and outreach or extension. The historical mission for BioMarc has been associated with a Regional Center of Excellence Award for Biodefense and Emerging Infectious Diseases. It was established in 2006 as part of this NIH award. Our historical mission has been production of high containment biologics focused on countermeasures for infectious disease threats. And so there's a bit of a biodefense theme on it, and it's CGMP manufacturer. CGMP is current good manufacturing practices. This gets you into the realm of producing materials that can actually meet FDA standards, and in some cases even EMA, which is a European Medicine Agency. These are the major regulatory agencies in the world for materials or medicines or drugs or biologics that go into humans. Being part of the Infectious Disease Research Center, we're, again, focused on infectious disease countermeasures. We work at BSL-2, BSL-3. We work with CDC select agents, as Bob mentioned, some of these including spore formers and even toxins. And when you look at our type of facility being able to do high biocontainment work for these biologics, in the U.S., there's only about 5 of these contract facilities in the U.S. So there's BioMarc. Texas A&M is bringing on a BSL-3 facility. I don't think they're online yet. There's one in the University of Florida Research Park that was funded by the government as a startup in Alachua, Florida. Emergent, I think, has one. And then there's also one at Fort Detrick that does both federal internal work for the U.S. Army as well as sometimes NIH, and then they also do selected work from outside entities. So we're nasty bugs in trying to make things that ameliorate or mitigate in these nasty bugs and the diseases they cause. Recently, since I joined, we've been trying to pivot into the cell therapy space. We've done some work with some equine mesenchymal stem cells. Haven't gotten a whole lot beyond that, beyond the initial efforts. But again, we're all contract driven from the outside. I already talked about how we were established. And again, this was a post 9/11 event. The federal government realized that the U.S. was somewhat deficient in biosafety level 3 infrastructure for research. and certainly for biomanufacturing. So as I was getting earlier, our base certainly is Colorado State University. We are out of the Office of the Vice President for Research, and moving up the pyramid here, we're part of the Infectious Disease Research Center and have BioMarc at the apex because we are the most important thing around. That's kind of a joke, but this was a figure started by our executive director of the Infectious Disease Research Center, so I'm going with it. If we look across the spectrum of research at Colorado State University requiring BSL-2 and BSL-3 containment, a lot of it, and I'm not even sure it's the vast majority, but certainly a lot of it occurs out at the IDRC, which is the Infectious Disease Research Center. We've got about 120,000 square feet, with almost 50,000 square feet of that being BSL-3 laboratories. Certainly biosecurity and biosafety are paramount. We have rigorous training programs in biosafety, biosecurity, and research compliance. Bob's team operates a very nice mock BSL-3 training laboratory. There's no agents in there, but it looks very real. And for the first several weeks of my tenure at CSU, I would go by there and look in, and there were some animal cages in there, which, you know, those happen sometimes in laboratories with little furry animals in there. And it was several months before I finally realized that they were stuffed animals and not real. So the mock training lab makes things as real as possible without taking any risk with anybody. Within the Department of MIP, or microbiology, immunology, and pathology, they have three major strengths, and those are associated programs in mycobacterial research, prion research, and then the vector-borne group. And the major themes are around countermeasures or vaccines, but certainly they involve diagnosis and therapeutics. And some of the agents that are worked with include TB agents, including multi-drug resistance and extended drug resistance strains, Yersinia, Francisella, Brucella, Burkholderia. So there's some pretty nasty bacteria right there. And then when we get into the virus, the whole encephalitis group, major ones being Venezuelan, Eastern, Western, St. Louis, West Nile virus, and Japanese encephalitis virus. Then moving down on through the list, we have Rift Valley fever, dengue virus, yellow fever virus, chikungunya, lacrosse, Sinbis, Powassan, and then getting into some of the others, we have work that has been done with MERS, ongoing work with high pathogenicity, avian influenza, and rabies. And then I bolded SARS because it seems like almost an extremely large component of the university research resources, including those at BioMarc, are being directed at SARS coronavirus type 2. So a little bit more on BioMarc. We're very unusual, and as I've hinted a couple of times, we're thought as a startup, but we're not really a startup because we're not really a company. We're an honest-to-goodness component of CSU, but in the same sense, we're also not really a research entity. We are very much driven at timelines and budgets and producing materials for clients. In that realm, though, we're a not-for-profit, and we are a contract development manufacturing organization. So some of these types of organizations are CMOs, or just contract manufacturing organizations. We spend an enormous amount of time in development because many of the projects are brought to us, and to borrow a euphemism, they're not ready for prime time. They're not ready for moving straight into a manufacturing effort. And so there's a lot of development that goes on, and that may be growing the virus to higher titers. It may be growing the bacteria to higher optical densities. It may be enhancing the production of a recombinant protein. or it may be associated beyond that in the downstream processing and the end of it where we talk about purification. We do specialize in high biocontainment. One thing that's really important for us is a phase of appropriate CGMP quality continuum. I'll talk a little bit more about that, but quality is a critical component of what BioMarc does. The materials that we manufacture may be for use in preclinical studies, that is in animal models or in vitro studies, or clinical studies, and we are primarily aimed at phase 1 and phase 2 clinical studies. So if you've been following some of the SARS coronavirus vaccine reports over the last several months, companies have moved from phase 1 to phase 2. We have several that are all the way in phase 3. And then once you move beyond phase 3, it's commercial manufacturing. We do very little of that. The next bullet point down, process and method development, is where we spend the vast majority of our time and effort. Ideally, we would like to spend more on that because the more someone invests in the early stages, the less it costs you when you move into GMP manufacture, which is, for the effort, is much, much more expensive. So it makes sense to spend more time in process and method development to get that really rigorous and robust before you move into GMP manufacturing. However, we have great difficulty in convincing our clients, particularly those in startup companies, because they usually haven't got the experience of moving something from concept to early research and development into process and method development and eventual GMP manufacturing. And so they don't understand that. They're in a hurry. We actually get calls from clients, and one of the earliest questions we ask when we start talking with them is, and what's your timeline? When do you need material? And I would say at least 25% of the time we get a response that's half in jest but half seriously. The answer is yesterday, which means they're in a hurry. They want to take shortcuts. They want to get to the phase 1 material as quickly as possible, which somewhat short circuits the effort in spending extra time in process and method development. We can bank cells and virus, that may be mammalian cells or bacterial cells. We can do stability studies on the material, and then we have a limited capacity for aseptic and viral fill finish for actual individual dose files to take into a phase 1 or phase 2 clinical trial. The vast majority of our work is in the area of vaccines. We've done a little bit of work in biotherapeutics, and then we've done quite a bit of work in diagnostic reagents and kits. We've been inspected by the FDA back when we were doing a commercial manufacturing project. We are regularly inspected by CDC, Homeland Security, and USDA, and that's associated with the Select Agent Program. And as I mentioned, we have taken an early foray into trying to get into stem cell work. Oh, that's interesting. So we got a bunch of animations that aren't going to work here, but that's okay. Yeah, I can't believe that. So the purpose that we developed in trying to work on mission statements for the organization when I first joined was to defeat global health threats. And the previous director, he and I overlapped for about a month and a half. One of his parting shot contributions was the tagline, "We do the difficult." And part of that is in reference to trying to do GMP manufacturing in a BSL-3 environment, because we're caught in a conundrum there because CDC wants us to keep everything out, but everything in to contain it. And FDA wants to make sure that we're making good, clean stuff, so they want to keep everything out. And so it takes a bit of coordination to get from the propagation stage, if we're working with a BSL-3 agent or a select agent, certainly, to get it to the point where we can transition it from a negative pressure environment into an FDA-compliant environment, which is usually positive pressure. As a group, our core values are honesty. Nobody likes to be lied to or have things misrepresented. We have a pretty broad depth of knowledge, and we are creative because it all the time is requiring us to develop creative solutions to challenges. We're proud of our achievement, and we believe that we're pretty highly competent. So one of the things that we have difficulty in explaining is we're a manufacturing organization in a university. There aren't very many of these. I mean, the Gates Center down at Anschutz is certainly another one. There's one at the University of Wisconsin, but most of, and then Texas A&M as well. Many of these, not all universities have these associated within the university framework. They are often in a spin out or an industry park, and then when we get into doing the difficult in BSL-3 biomanufacturing, that is really unique. So a couple of key points here. If I don't leave you convinced that moving scientific advances from the bench or the laboratory into an actual manufacturing is very hard, then I've missed my target here. And certainly getting that transition from research and development into manufacturing of a commercializable product, it's very essential that you are able to do that to have impact in the world. But associated with the basic science and the scale up are a bunch of regulatory issues, predominantly for us with the Food and Drug Administration. We believe that making all these efforts with our resources and expertise will make a difference, and that the challenge is worth our efforts. And in order to have a significant impact, and in order to impact the world, make it a better place, make it safer for people, solve a disease problem, we need to have, everybody needs help to get products to the market in use and mobilization out in the field. The biggest difference is stolen from a Ebola clinic clinician that I had met, and he's at University of Nebraska Medical Center in Omaha, and they had done a lot of pre-outbreak work in how they were going to handle patients. You may be aware that Omaha is one of the places where they have a clinical BSL-3, and they took some of the Ebola patients from the 2016 outbreak, some of the medical personnel that had come down with Ebola, and treated them there. So that's, one or two patients at a time is one thing, but when you start going into a village where they're having an outbreak and you have hundreds of people dying, big indeed is different. And for us, it means going from a petri dish level in the laboratory to a 200-liter bioreactor. That is a huge step, and so that's what I mean by big is different. So while we're part of the university, we are just like a company in the industry, and from that aspect, we have functional departments. We have some people dedicated to business development, that is working with clients and developing contract proposals. Key to our success is project management. There are just tons and oodles of moving parts in any given project. And so we have a group that is tasked with keeping track of the moving parts, keeping people coordinated, ensuring that materials with long lead times are ordered in sufficient time so as not to negatively affect project timelines. And they also are the major point of contact with our clients. And also associated with getting into making materials that are suitable for use in humans, whether it's a clinical trial or even commercial manufacturing, is quality. My quality assurance manager would beat me over the head if I didn't impress upon you that there are two main goals for BioMarc, and that's all there is. And one of them is patient safety, and the other one is product quality. And so we have a group that is dedicated to quality assurance. And some of you may remember the Ford Motor Company tagline a couple decades ago. It's probably 20, 25 years ago. Quality is job one, and that is absolutely the truth. Then we have another group that is associated with quality assurance, but they're also separate, and that's quality control, where they do a lot of analytical work in measuring various aspects about the product. I'll talk more about that. Then we have a group that is into analytical and process development, analytical aspects from developing the tests that quality control will apply, and process development, as I mentioned earlier, getting from that Petri dish level up to 1 to 5 liters, maybe 50 liters, and eventually into 200 liters, which is our particular max capacity. There are manufacturing levels for various biologic materials out there with commercial pharmaceutical companies that are as 10,000 to 100,000 liters is not unusual. We certainly don't have the ability to go that big, but once you get beyond 50 liters, you're getting into big. Then we have another group that is dedicated to manufacturing. So after we get our processes developed, then we transfer them into manufacturing, and that is at our 200 liter level if necessary. Sometimes we're at smaller levels. It just depends on how much material we need. And then surrounding all of this, even though I have them last, is our facilities and operations group. We have to make sure that all the facilities are operating, the instrumentation and equipment is properly calibrated, it's fully functional, it's been validated, and so it performs as expected, and they're in charge of that. So those are all departments, where I get to this next slide, those are all departments that you would find in any commercial manufacturing entity, either in animal health or human health. They're pretty much all the same thing. Might have slightly different names, but the functions are pretty much the same. So, as I mentioned, we are a contract-driven organization, and so we go from project to project. Some of these may be 6-month projects. Some of them may last 4 or 5 years. I don't think we have any longer than that. But they have a life cycle, and it takes time to develop a project. Initial client contact, sometimes we will have those when we go to meetings. More often than not, people are searching for BSL-3 biomanufacturing. They find our website. We come up pretty high in a Google search, and then they reach out to us either by email or a telephone call. And the next step usually is a non-disclosure agreement or a confidential disclosure agreement so that we can talk more specifically about their project. So we move into the project needs and developing alignment between BioMarc and the client based on their needs and what we have available with respect to services, and then timeline. As I mentioned earlier, we have some clients that honestly have been in such a hurry they wanted it yesterday. And so right now we're booked solid for at least a year, and if some of our contract options are picked up by NIH, we may be booked for as much as 5 years. After we get an alignment on what they need and what we can provide, we develop what's called a rough estimate, or relative order of magnitude estimate, or an ROM. That's to float by what we think it's going to take to accomplish their project. It takes time to develop these, because the last thing we want to do is underestimate things and get into a contract situation where we need twice as much money and time to accomplish the project as what we've contracted for. We're not for profit, but it's also been emphasized we're not for a loss either. So if we have good feelings between us and the client at step 3, we move on to step 4, where we do a deeper dive on the intricacies of the project and develop a formal proposal. If they like that, that moves on to a contract signature, and then quickly thereafter, depending on when we can fit it into our schedule, a project launch. The entire cycle from step 1 to step 6 can easily take 12 to 18 months. We haven't done very many of them in less than 12 months. And depending on the seriousness of the client, sometimes they're really interested in moving ahead quickly. Sometimes they're just kind of shopping around. So that falls into the 12 to 18 month range in time it takes to actually get going on a project. So this slide is a horrible mess because it's animation loaded. We are globally community engaged. Starting from the top will be the easiest. We have NIH. We have 2 active contracts with NIH that we just got this year, actually just in the last couple months. We had one earlier this spring with BARDA. So between the 2 NIH contracts and the BARDA, they are all focused on SARS coronavirus type 2. And we are pretty much have moved from other projects on other subjects, other agents into those. CEPI's in the background. CEPI is the Coalition for Epidemic Preparedness Innovations. They're headquartered in Oslo, Norway. They are a non-profit amalgamation of a number of charitable organizations, the Bill and Melinda Gates Foundation, the EU consortium, the government of Norway, and some others as well. They are focused on developing solutions for emerging infectious diseases. And so our project with CEPI is an investigator-initiated project. My team and I put together a proposal in 2019 for a Rift Valley fever vaccine. We were successful in getting awarded, and that one is active now as well. Over here, we have the C, and that's from the Centers for Disease Control. We have a project where we're involved in manufacturing yellow fever diagnostic kits for them. Over here on the left is Boston Children's Hospital, which is part of Harvard Medical School. We're working on a vaccine for pediatric typhoid in underdeveloped nations. We're also working on an HIV vaccine, and I think that pretty much covers what's in the background of the slide that you can't see. I apologize for that. I think that's the worst one of them all. So to talk to you a little bit more about project management and what it entails, and I'm going to try and move quickly through this because I'm running a little slow. Project management is involved in developing the schedule, identifying milestones and delivery. We're very much timeline and budget driven, resource evaluation, materials review and planning, and the scope of work. So this is all work that goes on during the contract negotiations or putting the formal proposal together. After we get a contract, then PM is the designated point of contact between the client. We don't want them calling me or the business development people or the technical director or the quality people. They all go through the project management individual. That way there's no question on who to contact. And then we have kickoff meetings where we plan and coordinate how we're going to do things. A lot of moving parts, a lot of coordination. I like to euphemistically refer to our project management group as the hub of the wheel, with spokes going out to the different functions and the rim of the wheel being the whole of the project. They're involved in budget management, managing the timelines, and we use planning tools like Gantt charts on Microsoft Project. They are proactively in recognizing and mitigating project risks, so they lead a risk analysis where we identify all the different things that could go wrong with the project and how we're actually going to mitigate those issues before they arise and how we'll manage them if they do. Then they facilitate in the project process at the operational level. So as I mentioned, they're the hub of the wheel, and they're essentially the overall project coordinator. There's a lot of conference calls. Some of our clients we speak with on an every other week basis, others monthly. Sometimes there's communications in between. but we usually have a PowerPoint presentation that we'll prepare and share with them prior to the meeting. We'll go over that. We'll update action item lists and discuss whatever issues and future directions, and they'll develop minutes. And it's very important to have minutes as well as the action item issues decision log, because you'd be surprised how much people forget what was said in the meeting, what they committed to. Oh, I don't remember that. Well, here it is. It's in the meeting, and it in the minutes. It helps ameliorate prolonged discussions and disagreements based on somebody not recognizing that we'd already covered that particular point. We have project reports. We have technical reports. We have schedule status. We update the risk register, and we look at the status of the deliverable. So there's timeline, there's budget, and the ultimate is the deliverable. What is it that the client wants? And it's always nice if we can get that done ahead of schedule, but we seem to live in the land of Murphy's Law, where if something can go wrong, it will. And so that's why the risk register is really important, but we want to communicate very clearly with the clients where we are and what we see coming up. And that gets us into a percentage of technically complete on the particular project. Project management has a monumental job because they serve as an advocate for the client and also for the BioMarc project team. So they're essentially caught in the middle. And people who are successful project managers develop some of the best negotiating skills that you've ever seen, as well as being able to communicate clearly and with a bit of tact. So their key thing is understanding and managing the project scope. So it's one thing to understand, but to manage and to articulate as well as coordinate all the different moving parts of a project is a very important aspect. A really good project manager, and we are fortunate to have two excellent ones, can make or break your entire effort when you start getting into these complex projects, as well as multiple complex projects running at the same time. So getting into our quality system, we work on a 6-system approach, and that involves production systems, facilities, and equipment. And you can see this is where some of our actual functions come in. Laboratory control systems, material systems, and eventually when you're making a product, the packaging and labeling system, but this also gets into intermediate material tracking. We operate under what we call a CGMP continuum, and that'll be a little obvious in further slides. But generally, we're making material for preclinical development or research and development. Cell and virus banking have their own set of FDA and industry guidance regulations associated with that. And then moving from preclinical into phase 1 is a big jump in quality requirements. And then from going from phase 1 to phase 2, yet another increase in quality development. So our continuum is we give enough quality to satisfy and meet the requirements for the particular level instead of taking a commercial level quality system, which is the max and the ultimate and absolutely the most expensive, and applying it across for all of these. Some contract manufacturing organizations actually do that. It makes it just god-awfully expensive, and more so than it needs to be. So, quality provides leadership, oversight, management of those 6 quality systems. As a matter of fact, our quality assurance lead trumps any decision I will make because they are the ultimate decision maker as regards to quality of product coming out of our laboratories. These systems comply to regulatory standards, and as I mentioned, we're almost exclusively FDA, and it is appropriate for the phase of production, and it must adhere into our internal procedures, which are somewhat developed around the regulatory standards. So they provide training. They do a lot of auditing, so that's how we actually manage our quality. And there's a lot of accountability. So if something is not right, it's not a matter of really pointing out, trying to find out who was wrong, as it is finding out what went wrong and making corrective measures so that we don't have that problem again. So we have a quality manual. We have standard operating procedures that are far more rigorous and intense than you might see in some of the research laboratories at your various institutions. And when we're going into actually clinical material for phase 1 or phase 2 studies, we often will enter into quality agreements with the sponsors, particularly if they're a large biopharma entity. They have their own quality teams, and we'll enter into a binding document called a quality agreement. Our system is paper-based. We are moving some of it into an electronic database. When you get into big pharmaceutical manufacturing systems, they're electronically based. We are focused on continuous improvement. So as I was mentioning in the previous slide, if we have a problem, it's not so much to punish somebody who made a mistake. It's to find out how that mistake was made and bring into account corrective measures so that we don't make that mistake again, therefore continuously improving. And again, in our CGMP quality continuum, we comply to the regulatory standards appropriate for that particular phase of production, whether it's preclinical, phase 1, or phase 2. And so here's another way of looking at the quality continuum. So if we're started here at the left, this is research and development. This could be in an academic laboratory. It could be in a small biotechnology company, startup company, but it's the pre-GMP phase, pre-good manufacturing practices phase. So they can make material that's for preclinical studies, usually a toxicology study, and there we usually apply good laboratory practices. From the very beginning, we apply what's called GDP, or good documentation practices. And then somewhere between the preclinical stage and phase 1, we start the PV, or process validation life cycle. So as we're developing the process, we want to start developing a process that is robust and repeatable, so that whenever we do it, we have a certain range of expected yield, and we can have high confidence that every time we try that, it's going to have a yield within that range as long as everything was done properly. Also, what happens in this phase is scale up. So we're going from Petri dish level here to moving into, if our max volume is 200 liters, moving up to 200 liters. It's good and best to have calibrated equipment at this phase, but certainly by the time you get to phase 1, you do have to calibrate and qualify all your equipment. The analytical methods move from qualification, that is, are they suitable, scientifically suitable for application, to full assay validation, which involves multiple operators over multiple days, pretty much getting the same thing day after day. And these are all involved in increasing GMP expectations, moving from phase 1, phase 2, phase 3 clinical trial material, eventually into the commercial phase where you have fully validated process plants, and it's all dedicated and controlled as much as possible to have this.
You need to unlock your John's iPad first.
Then getting into bioburden and endotoxin control. So we do a lot of injectable materials, so you want to make sure that they're sterile. And then process validation. By the time you get into commercial, it's fully validated. You have the most that you're going to get as far as process, product, and CMC, or chemistry, manufacturing, control, the knowledge and understanding of that. So that's why quality is very important in manufacturing. And these sort of principles certainly apply to any pharmaceutical that's going into humans, whether it's a biologic or a straight-out drug. We have to be inspection ready. Main point here is we had a pre-approval inspection in November of 2013. The Form 483 is what the inspectors use to document the findings that require corrections. We had 11 findings on the Form 483 in 2013. They came back and did a surprise inspection in July 2016. We had no Form 483 findings. So that is a very, very good performance. So in our quality control labs, we focus on safety. And again, a lot of the things, the biologics that we are manufacturing are injectables. So we have cytopathic effect infectivity testing. We assay for bioburden, which is a little bit lower standard than absolute sterility. Endotoxin from bacteria, because these can cause adverse events. And then checking also for mycoplasma. These 4 tests that are in parentheses are outsourced to commercial laboratories because they run them day in and day out. And it also brings in an independent outside source to give more veracity to the results. Then we focus on identity. So is the material exactly what we think it is? There's a couple of analytical methods that we can do here, looking at molecules and immunogenicity, and then looking at some of the nucleic acids associated with that, whatever the particular product is. Like I said, we deal with viral vaccines and also bacterial vaccines. Strength is also very important. So if it's a live virus, we look at infectious titer. We can look at antigen concentration through ELISA. We can look at RNA copy number. So it's all getting into a quantitative measure of the material. And then purity is very, very important. Whenever you're growing a virus, when you first harvest that viral infection off of the bioreactor, there's going to be residual host cells because we have to grow viruses within a cell, a eukaryotic cell. So that can cause adverse events. We use benzonase a lot to digest host cell DNA that comes in. And so we have to look at protein impurities because if we add benzonase in, then we have to purify that out. But you have difficulties getting 100% of that. And there's standards that FDA has for maximum amounts of some of these things. And then again, the host cell DNA assay, how much host cell DNA is contaminating your product. And then getting all the way back into quality. So appearance, pH, molecular components, total protein, all of these get into product quality control testing. We also utilize the core facilities at CSU. I won't go into all of them, but we have a proteomics and metabolomics group, and we have availed ourselves to the mass spec there. The central instrument facility has a number of things. We'll be engaging in some transmission electron microscopy soon on a couple of our viral vaccine projects. The microscope imaging network. One of our collaborators on the coronavirus vaccine project has been doing quite a bit of flow cytometry, and then we're also using our next generation sequencing and genomics center. The veterinary diagnostics group is also involved with the experimental pathology group looking at tissues in some of the animal models that we've been working with, again, on the coronavirus vaccine project. So there's a cycle in quality control testing. So you initially can develop a method or bring a method in through a technology transfer from the client if they have those. You will perform a method development. You'll go through a design of experiment exercise. You'll establish the method operable design region. So what is the tolerance zone of expected results there? You'll verify it. You'll validate it. We use system suitability controls throughout our assays, and so that we can have, it's not really a control, but it's a reference. And so we'll do continuous monitoring and develop method performance requirements, and we'll refine those. So this is a continuing cycle here of quality control testing, and that's really important. And these are applicable to different levels based on the phase of material that we might be producing, whether it's preclinical, phase 1, or phase 2 clinical material. And I'm not going to get really into this. We do apply quality by design, that's QBD, in our analytical early phase, but we also do quality by design in some of our actual production methods as well. And so these are all designed through good manufacturing practice regulations and guidances to have analytical tests or processes that are robust and repeatable. That is extremely important in manufacturing. Here's another look at the different aspects of analytical method, and it's also a continuum from the preclinical phase, moving to phase 1, phase 2, phase 3, and eventually commercial. So initially, you can have an assay that is scientifically sound, but eventually you need to be moving into qualification. And then finally, when you get closer to actual commercial production, this has to be validated. And so the intensity of work on the assay and refinement of the assay increases, and that's why the arrow goes from light green to darker green as we move down. And the same for these other aspects. So we have regulations like 21 CFR 211 that talks about suitability for the intended use, and this gets into identity, purity, safety, and the quality of the material, and how the test method actually evaluates those aspects. And again, as you move further along during the development phase, from preclinical, phase 1, 2, 3, and commercial, the requirements of all of these aspects becomes more intense. And so that means more time, more effort. And eventually when you're talking to a client, it's more expensive. And that's why I was saying we operate on a continuum so that we don't give commercial level quality that is the most expensive when we're really just developing preclinical material for a client. Our facilities and operation group. So this is an older aerial view of the Infectious Disease Research Center. We have a new building that is nearing opening construction back here now. And this houses our vector-borne diseases group, or will. I think we're having grand openings soon. We have 2 different CGMP sites within the Infectious Disease Research Center. Our biosafety level 3 labs are found in this wing and this wing. This portion is almost entirely occupied by BioMarc. We have a number of, 4 different production or separate BSL-3 laboratories. Here we have, and so we can upgrade them to, they're BSL-3 capable. We can downgrade them to BSL-2 depending on what we need. And so we can manufacture preclinical material there, as well as drug substance or active pharmaceutical ingredients there. So we have manufacturing capability. Here we have a BSL-3 analytical lab over in this section here. And then this building here is called our GMP production building. This is total BioMarc space, where the rest of this space is shared across the other investigators at the Infectious Disease Research Center, the vast majority of those being members of the microimmunology and pathology department. And then we have some more BSL-3 capability over in here. So the GMP building, as we call it, is BSL-2 only. That we have our main quality control lab is in there. We have office space, and then we have 2 tandem production suites for a total of 4 in here. And so we can do manufacturing for preclinical drug substance and active pharmaceutical ingredient, as well as DP or drug product. So we have our formulation and filling suite in here, and we do a semi-automated or mostly manual type of vial filling, and so our capacity is about 1,500 to 2,000 vials at any given time, which is sufficient for phase 1 and phase 2 clinical studies. Getting into phase 3, you're going to get into tens of thousands. So that's one of the main reasons we focus on preclinical phase 1 and phase 2 materials for our clients. We are part of the Rocky Mountain Regional Biocontainment Laboratory, and again, this facility was in large part constructed through NIH funding. We occupy about 9,000 square feet, and in that we have BSL-2 and BSL-3 non-GMP space, because our spaces, as they're controlled for GMP manufacturing, there's a stark difference between non-GMP space, which is for the preclinical material, whereas the GMP space is for our phase 1, phase 2 clinical material. Then we also have, as I mentioned, a BSL-3 quality control lab. We have offices, and then looking at our GMP manufacturing building, it's about 8,500 square feet. We have BSL-2, it's all BSL-2, so we have BSL-2 GMP production space as well as our formulation and filling suite. We have our BSL-2 quality control lab, and we have offices there as well. A little bit about our development and production spaces. We use exclusively single pass air, and we have HEPA filtration on the supply air as well as the exhaust air for the BSL-3 labs. We have full emergency backup electrical power. We have a number of different, I think we have 3 very large diesel generators, diesel power generators on the IDRC site. We do clean room commissioning in accord with our BioMarc standard operating procedures, and these will vary based on the process and the phase, whether it's preclinical, phase 1, or phase 2. They all have different requirements, and they're increasing stringency from preclinical up through phase 2. So equipment management is very important, as I mentioned, calibration, and then equipment verification. Classification, is it GMP or non-GMP? Tracking where these are with these different production suites, whether they're non-GMP or GMP, we are constantly moving equipment in and out based on what is required for the projects. They're in that flexible approach. It's also phase appropriate, and they're project specific. So we don't have all suites fully equipped with bioreactors and perfusion skids and tangential flow filtration or diafiltration. We equip each space as required. The basic things that are in each of the suites is an autoclave, a pass-through autoclave, and a biosafety cabinet, and then in a sink. And after that, it can vary depending on the project, whether we have refrigerators, freezers, purification equipment. Just depends on what we're doing. So it's very important for us to track our equipment. Technical capabilities. This is kind of a busy slide. Here is a picture of a Sartorius STR200. This is a 200-liter capacity single-use bioreactor, and so these are a plastic-type bag. They have fittings for various hoses, and we can pull off culture material. We can add media. We can add cells. We can add infected cells. We can monitor gases such as oxygen. We have the capacity to measure temperature, pH, glucose consumption. Just depends on the process that we're doing. And these 2 stainless steel doors on each side kind of clamp around the single-use bioreactor. It also has its own magnetic drive impeller that goes down through here to move the culture around. Each one of these bags, depending on how many ports are customized into here, are anywhere from $2,000 to $5,000 each, and they're single use. So you use them once, You drain them, get your material out, you compact it as best you can, bag it up, and autoclave it, which makes cleaning very, very nice. So clients, they vary from virtual startup all the way through to large pharma and government entities. We do a lot of technology transfer, a lot of process and method development, engineering studies, in-house testing, GMP manufacturing, all of these things. And this is kind of a client introduction slide. So one of our business development guys added this nice one, taking your product from concept to creation. This is a bit of a repetition from before, but we are capable fully of CGMP at BSL2 and BSL3. Associated with it, we have collaborators with animal biosafety level 3 facilities. Dr. Bowen, who you heard from earlier today, is a big collaborator of ours on the coronavirus project as well as the Rift Valley fever vaccine project. We have experience with TB organisms, Clostridium spore formers, as well as Clostridial toxins. We've worked on a number of viral systems, including alphaviruses or the encephalitis viruses, flaviviruses, HIV as a retrovirus, and we are select agent approved and have some of our labs are actually select agent stages. We spend the vast majority of our time in talking with clients and trying to help develop their project to convince them that the extra time and money invested in process and method development will translate into shorter timelines and better product when we can move into GMP manufacturing. But we can grow bacteria, we can grow cells, we can do anaerobics or aerobes, a number of different virus infection models, as well as the mammalian cells to grow the viruses. For purification, we use almost exclusively chromatography or filtration. So filtration could be either ultrafiltration or diafiltration. And consideration and continuum in process design. So when we start taking a project in and scaling it up, we are doing everything with the concept of taking this project into actually commercial manufacturing. And so that is a little different from just growing 10 5-liter cultures or 10 1-liter cultures. Going to 200 liters is an arduous process, and it needs to be done right. Some of the equipment, we have a number of bioreactors. They vary in capacity from a half a liter to all the way to 200 liters, the biostat, Sartorius biostat. We have wave bioreactors, which are a rocking type, bag type thing. Purification, we have a number of ACTAs from the relatively small scale purifier model, which is 100 mils a minute, to the Avant 150, it's 150 mils a minute, to the ACTA Ready, which is more of an industrial size. So we're talking at liters per hour, 500 liters per hour with a high flow kit. So again, this is all geared towards actual manufacturing. We also have the MaxSite, STX, and VLX transfection systems, and we have the capacity of going from 100 microliters to a full liter on those, and that's really important in some production systems. In trying to pick up the theme of the meeting, we do operate fully as a part of CSU through the CSU compliance functions. So we're constantly in contact with biosafety. All of our projects are run through the Institutional Biosafety Committee. So we fill out ARFs and PARFs on each project. The ARF is an agent approval request form. The PARF is a project approval request form. And that gets at what are we bringing in? How familiar are we with it? Do we have the appropriate knowledge in handling this material? the proper PPE, the proper disposal, and what spaces are we going to use these agents in? What are we going to be doing with them? What scales? Because in general, if we take a BSL-2 organism, once we hit 10 liters in production volume, we generally are increasing the containment level to a BSL-3. We also coordinate with biosafety on moving our materials out of BSL-3, and in particular shipping some of these as hazardous materials off-site. We're involved with the Environment, Health, and Safety group for chemical ordering and inventory, hazardous waste disposal, and one thing I didn't get in there was the respiratory protection program. And then we're also highly involved with RICRO, which is our Research Integrity and Compliance Review Office. They have actually two quality managers now, and this is primarily for GLP and moving into human clinical studies if we're ever involved in those. And then there's also export controls, some things or technologies, materials, biologic agents we cannot send to certain countries. So that's kind of how BioMarc operates within the CSU system. I thank you very much for your time and attention.
Thank you, John. Really appreciate that. And we ran a little bit over, so I think, Denise, I'll turn it back over to you. And if you want to take a couple of questions, if there are some, that's up to you. But otherwise, we'll thank John for his delivery and for all this information he's given us, and also very much appreciate what he is doing for the biologics that we need to get out as products.
You know, Bob, I think that we're gonna be able to start the round table, and if we're a few minutes late, that's okay, because we have a break at the end of that, and we can truncate our break to accommodate the panel. I'd hate to see, if we've got any questions, I'd like to take advantage of Dr. Wyckoff's being here and could answer any. So, Cynthia, do you see any questions?
I am not seeing any questions right this minute.
I see some in the Q&A section. One is,
oh, there you go. I just popped them up.
do you have external customers work in your facilities, or only direct the research of your staff? So your clients, are they working in your facility as well, sounds like?
Generally, no, we don't have clients in our facility. We have had that in the past, but we haven't had that in the past 3 years. And it's generally for technology transfer of something that is along the lines of you've got to hold your mouth right and stand on one foot when you do this to make it work. So we've the ability to do that, but it's not a very regular practice at all.
And I cannot see the question, so Zach or somebody else, if you'd go ahead with those, that'd be great.
There's one more, and it was, how do you reconcile scheduling issues with employee health and safety concerns in the lab? Seems like there may be a lot more scheduling pressure, which can lead to working tired, stressed, and be more prone to accidents.
That's an excellent question, because it's very pertinent and timely. We are limited still by the governor's mandate that we try and keep staffing at 50% or less. We have a lot of paperwork that's associated with the quality program, so a lot of this and a lot of our training can be done online from home. But in trying to schedule activities and maintain social physical distancing in laboratories or in the office cube spaces, it's a very big challenge. And right now it is impacting our productivity and our efficiency. So that's a big problem right now, or challenge. As far as tired people, we try not to have people in the laboratories for any really extended length of time if we can avoid it. So I don't think we're seeing any of that. Most people are working short stints in the labs.
Okay. Well, again, Dr. Wyckoff, thank you so much for a great talk. I know I benefited from it and learned a lot about just GMP and moving through the different phases. So that was very informative for me. So thank you.